All right, so we're going to pick up here with our next part, talking about that inner layer of our eye, that inner layer specifically the retina. All right, so we discussed previously that that inner layer can consist of kind of two general parts. There's a pigmented layer, and then there's the neural layer. Okay, of this inner layer of this retina. So here is our pigmented layer. All right, this layer right here. This is our pigmented layer. It contains these pigmented epithelial cells that contain melanin and other pigments to gather light. And then this whole section, kind of in this region here, this is all of our neural layer, all right? And the neural layer contains and is composed of three general types of cells. There are photoreceptors, there are what are called bipolar cells, and we have cells that are called ganglion cells, all right? So first off, to start, the photoreceptors. So the term photo, so photoreceptor, it's receiving and being sensitive to a photon. So a photon is a package of light, just one unit of light that is coming in from the environment over here, right? So these are our rods and cones, right? You've probably heard of rods, you've heard of cones. These are sensitive to different types of light. So some rods are sensitive in very dim light, low light environments, whereas cones are sensitive to high intensity light and can also differentiate and distinguish different types of color. Okay, we'll see the structure of rods and cones in a second. We also have a layer known as the bipolar layer. Okay, so the bipolar layer are these green cells that we see right here. So if you recall, when we discussed in previous lecture different types of neurons, we talked about multipolar neurons, bipolar neurons, and unipolar neurons. So our bipolar neurons, we said, were involved with some of our senses, so like our sense of smell and sense of vision. So a bipolar neuron has a single cell body, right, with a nucleus, and it has projections kind of coming off in either direction. So there's a side coming off here. We have all of these projections. So there's two poles, right? There's a pole here, and there's a pole here. So you call it a bipolar neuron. This is the middle cell layer of our neural layer, right? So we have photoreceptors, bipolar cells, and these ganglion cells. This bipolar cell is in the middle. It makes contact with the photoreceptors here at the synapse, and it also synapses with what are called our ganglion cells. So our ganglion cells is this last layer. It's the innermost layer of the retina, and it is what contributes to the inner lining of the retina with these axons that continue down. So the axons of all these ganglion cells kind of fuse together, and as they run and they fuse together, they ultimately leave the eye at a region known as the optic disc. Okay, so the optic disc, if we look up at this small image of the eye here, it's this general region kind of right here. I kind of circled this large, but it's this region right there, right as the retina leaves the eye and becomes the optic nerve. That's called the optic disc. Sometimes the optic disc is referred to as the blind spot because we contain no photoreceptors there, all right? So as we have our retina and it leaves with these ganglion cell axons to the optic disc, to the optic nerve, they're generating action potentials along the way. These action potentials will travel to the thalamus, where we then relay information, ultimately going to the occipital lobe of our brain, where we have our primary visual cortex, and we're able to interpret and perceive what it is that we are seeing. Okay. All right. So if we look here, we can see these layers. We can see the layers of this retina. So we have our pigmented layer most deeply, right, with those epithelial cells that contain melanin and other pigments to help absorb light. We have a photoreceptor layer here. We have a bipolar layer here, and we have our ganglion cell layer here. And remember, all of these axons of these ganglion cells fuse together, and they form this nice inner layer of the retina, and then to the optic disc right here, and then to the optic nerve back to the brain. Right? So we will look at each of these layers in a little more detail, and we'll start by looking at our photoreceptors, so our rods and our cones. So when we look at our photoreceptors, we look at our rods and our cones. We have two cell types here, these photoreceptors that are sensitive to photons, and they're named according to their general shape. Okay, so rods appear to be more rod-like. They're kind of more boxy, more rectangular, whereas cones are somewhat more conical in shape. So they have a more kind of triangular, pointed appearance to them. All right. Now, both rods and cones have some similar features. They're both composed of what's called an outer and an inner segment. They both have a nucleus, so they have a cell body, and they both have kind of a synaptic terminal, okay, which forms a synapse, which forms a connection with our bipolar cells. Okay, So whenever we're looking at the outer membrane, or this outer segment here for both of these, you see these stacks, or these kind of folds in this membrane here. These folds, or these what are called these discs here, they contain photopigments. So they contain these pigments that can be sensitive to light and can undergo changes in the presence of light. Okay, so we'll see that rods respond to low light, kind of dim conditions in terms of light or the amount of light, whereas cones are responding to more light, right? So a higher intensity light. But the way they kind of act is very similar, okay? So we'll talk about a pigment called rhodopsin, okay? So we see this term here, this rhodopsin molecule, which contains a pigment portion, and it also contains a protein portion. These are found within these discs that are stacked together. So these discs that we see here that are absorbing light coming from our environment. Okay, so we see this in both the rod and the cone. In our inner segment, this is where we have the mitochondria and other organelles associated with these cells, right? So our cells are, their photoreceptors are cells just like any other cell. So they have mitochondria, they have ribosomes, they have all these other things. We'll see they have mitochondria because we need ATP for some of these processes that we're going to talk about. They both have a cell body, right? So they have a cell body with a nucleus. So here's the cell body of the cone, and here's the cell body of our rod. And then they both contain a synaptic terminal here that will join and form a synapse with our bipolar cells, and they will release neurotransmitters into these synaptic clefts. Okay, so what we'll see is that depending on the type of cell, so we'll generally talk about it with respect to rods. Depending on the type of cell and depending on whether or not we have light or no light, we'll have different states. So we'll have a dark state or a resting state, then we'll have an active state, or kind of a state when there's light present. And the activity of the cell is kind of counterintuitive. So if we're talking about rods, whenever we have no light or we have very little light, so we're in the dark, we're in this kind of resting state, we have what's called the dark current. So there's sodium flowing into this cell. We generate graded potentials that flow down the cell. When they flow down the cell, they release all of these neurotransmitters down here that we kind of drew down here at this synaptic cleft, and that actually inhibits the bipolar cell. Okay, so in this case, there would be no action potential going to the brain. Whenever we have light, the opposite happens. So there's fewer graded potentials, which releases less neurotransmitter down here, and the release of less neurotransmitter actually stimulates and excites the bipolar cell. Okay, so that tells you that this neurotransmitter that we're talking about, this glutamate, is actually an inhibitory neuron, or an inhibitory neurotransmitter. So if you have more of this neurotransmitter that's inhibitory, it's actually going to inhibit the next neuron to not send action potentials. But if you have less neurotransmitter release in the presence of light here, you actually stimulate the next neuron, so you stimulate that bipolar neuron to then send more action potentials. Okay, so it's somewhat counterintuitive, but if you think about it, and you think about this neurotransmitter called glutamate and how it's inhibitory, it makes a little bit more sense. Okay, so if we look specifically here at our rod, so we look at our first type of photoreceptor. These are going to be our rods. Now our rods are far more numerous than our cones, so we have many more rods in our eye than we do cones. But we'll see in a little bit that we have more differentiation among our cones. Okay, but these are going to be sensitive to low light. Okay, so they're very sensitive to light. So there doesn't have to be a whole lot of light to stimulate our rod. So these can be activated even in dim light or low light conditions. So like at dusk or something like that. Okay, in the dark. However, these are not extremely sensitive to color. Okay, so this means that we can detect shapes sometimes, so we can see things, but we generally don't have very good edge detection, so you can't really see the specifics of an object or a shape. You can't really differentiate lots of features of it, and you can't see color as very well as very good either. So this, you know, this when you're in a dim light environment, you can make out kind of the silhouette of things, you can see general things, but you can't really tell a whole lot about it. You can't see many colors associated with that with that object. So if we look here, we see our rods. These are located more peripherally in the retina, so they're located more on the side here. So we see this in this image to the right as well. And you notice that they're in a gray-black tone. So this is telling you that they're not extremely sensitive to color. All right. So if we move past our rods and now we look at our cones, we know that our cones are sensitive to color. They're also stimulated by high-intensity light. So there has to be a lot of light present in order to activate and stimulate these cone photoreceptors. Okay. So they also allow for color vision. And we have 3 different types of cones that are sensitive to different wavelengths of light. Okay. So we have ones that are called green cones, we have ones that are called blue cones, and we have ones that are called red cones. Okay. Or maybe I already said red. We have red, we have blue, and we have green. Okay. Now the cones themselves are not red, blue, or green, but the wavelength of light that they are sensitive to, or most sensitive to, are either red, blue, or green. So that's why you see here in this one you have some blue ones, right? You have some red ones here, and you have a green one. So the color of the cone cell itself is not red, blue, or green, but the wavelength of light that it is sensitive to is red, blue, or green. We have the most red cones that are sensitive to the wavelength of light that is red. We have second is our blue cones, and then green cones. But the most, so somewhere around 70–75% of our cones are red, and then around 15 or 16% are blue, and then about 10% are green. Okay, so the vast majority are red, and you can see this down here in this image. You see lots and lots of red, you see a little bit of blue, and you see a little bit of green, but by far you see the most red. So our cones are also allowing for very sharp, clear vision as well. So we have a region here known as the fovea centralis that we circle right here. So this region right here. This is our area of best central vision. This is where we have the highest acuity, the best sharp vision possible whenever light is focused on that area right there. Okay. All right, so let me clear this here. All right, so you may have heard of color blindness, right? So speaking of these cones, you can have color blindness in which you have a deficit in one or more of these different cone types, all right? So color blindness, the most common type is kind of red-green colorblind. It is what's known as an X-linked recessive disorder, okay? So if you think about the chromosomal makeup of biological male, biological female, you have, and this is in the vast majority of cases, males are XY, females are XX. So what this means is X-linked recessive. So recessive meaning it is not the dominant allele of this gene. It's not the dominant form, but it's the recessive form. So if it is X-linked recessive, this means it's carried on the X chromosome. Okay, so both men and women can be affected, right? Both biological male, biological female can be affected. But it turns out that males are more commonly affected, okay? So think about why, right? So answer this question, why are males typically more susceptible than females? You would think, okay, females have XX, they have 2 X's, so they're twice as likely to get it, whereas males only have 1 X, right? Well, it turns out it's exactly the opposite. So because males only have 1 X chromosome, if they inherit just this one recessive gene, then they're going to show it. Then they're going to be colorblind, okay, for this specific type of colorblindness, right? If a female receives the recessive gene on 1 X chromosome, so say she receives it on, if we're looking at this example here, she receives it on this one, right? She receives it on this X chromosome, but this X chromosome is normal, then she'll express the dominant allele. So she'll be normal with respect to her color vision, right? And so it actually turns out that males are more likely to show or to have color blindness because they only have the 1 X chromosome. So if they receive just one recessive, this one recessive gene, then they're going to show color blindness, okay? So you can look at this image up here, and hopefully you can make out the number. So this is 74, right? So if we color it here, you have a 7, and then you have a 4 here. If you happen to be red-green colorblind, then you could not differentiate all the red dots from the green dots, and this would just look like one circle of smaller circles, right, of smaller dots. And so this is the most common in which red and green are most difficultly, it's most difficult to differentiate between these different colors. Okay, so again, this is having to do with the cones in the eye. Okay, so when we have these cones concentrated in a specific area of the eye, this is known as the fovea. Okay, sometimes known as the fovea centralis. It's in the back of the eye, the back of the retina. And we have a very high concentration of cones in this area. So if you think about what cones do, what they're sensitive to, and what type of vision they allow, then this is an area where we have that best vision. So we know that they are good for color vision. They're good for very sharp, clear vision. So in this area right here, this fovea centralis, this is where we have our highest visual clarity, highest visual acuity. Okay, so lots and lots of cones. It's only cones, so you can use this mnemonic here. So fovea, an O, only, and then cones here with an O. So we only have cones in the fovea. Okay, and again, this is where we have very sharp vision, very good color vision specifically. Okay, so when we start looking at the pathway, so this pathway of bringing in light, stimulating a cell is known as phototransduction. Sometimes it's referred to as photoreception, phototransduction, or this phototransduction cascade. Okay, so it's termed a cascade because there is a cascade of events that must occur in order to stimulate our cells. Okay, so if we're looking at this example here, and we'll look at further images and further examples to make this make a little bit more sense, but we're looking here at a cone. All right, so we're looking at a cone here. So whenever we have what's known as the off pathway, we're dealing with dark light, okay, or a lack of light. Okay, when this is present, when we're in this condition, our cell is depolarized. Okay, so in this pathway, whenever we have light present, it becomes hyperpolarized. Okay, so think about whenever we did excitable cells and we talked about action potentials, what is hyperpolarized, what is depolarized? Remember, depolarized, we're becoming more positive in the voltage across the membrane. When we hyperpolarize, we become more negative. Okay, so like I said previously, this idea between when we stimulate the cell with light and the graded potentials and whatnot, it's kind of counterintuitive to what you would initially think. So whenever we have light, we become hyperpolarized. That means we move away from threshold, right? We're getting more and more negative. So because of that, we send fewer graded potentials down this cell, so down our cone here, which means we release less glutamate. Okay, so if we look at this section right here, we're releasing glutamate in smaller quantities. So decreased glutamate is released. Okay, because you release less glutamate, you actually end up stimulating your bipolar cell. And when you stimulate your bipolar cell, this then travels, the action potential travels and stimulates your ganglion cell. And then when you stimulate your ganglion cell, you have an action potential going along the axon of the neuron to the brain. And so it's kind of opposite as to what you would think. And you could think of the same example, but instead of using cones, you could use rods. All right. So if you remember that the glutamate neurotransmitter is inhibitory, then you can think about what is happening at that synapse when there is more glutamate released or when there is less glutamate released. So if there's more glutamate, that's inhibitory. That's going to inhibit the cell. If there is less glutamate, that's actually excitatory because there's less of an inhibitory neurotransmitter. All right. So this is a video that you can watch that shows some of the activity of light on these different photoreceptors. So here we're showing a close-up view of the neurotransmitter release onto the synaptic terminals. Here we're looking at a rod in particular in one of those discs, and we'll talk about this in a few of the next slides. We'll look at the rhodopsin protein. Okay, this rhodopsin molecule. All right, so you can watch this. So here you see our opsin and our retinal. These are the two main components of this molecule. So you see the retinal in the middle, and then the opsin more on the outside. You zoom in on the retinal. So this is when it is in a kind of relaxed form, which is known as the cis form, the 11 cis form. Right? When a photon of light hits it, it'll actually change shape. So there you see it change. And now it's called the trans form. Okay, so it goes to the all trans form, right? So we reoriented some of these molecules here out on the side, all right? So we'll move past that and we'll look at this retinal epithelium and how light is stimulating it. So remember we said that light is actually coming through the neural layer, right? So we have light here. It's coming. So here's our ganglion layer, right? Here's our bipolar layer here. Here's our photoreceptor layer, and then here's the back of the retina way back here. So light is actually passing through this way, hits our pigmented layer, and then turns around, and then the signal goes back in the opposite direction. Okay. So in these epithelial cells, these pigmented epithelial cells, they contain pigment granules, so they contain melanin that we see right here, that can actually trap extra light and prevent it from scattering. And if this scattering were to happen, it would more or less kind of blur our vision. It would kind of obscure our vision and make it not quite as clear. Okay, so they can also store and recycle vitamin A, and we'll talk about vitamin A in a little bit and how it's necessary to form the retinal pigment in our photoreceptors. Okay, but again, keep in mind this pigmented layer is at the very, very back of our retina, and light has to pass through all of these other layers before it gets to that pigmented layer. Okay, so we're going to stop here, and we're going to pick up talking about receptive fields and probably finish in the next part.